thanks so much for having me. I've actually, amusingly, as much as I've hosted events here, I've never given a talk in this room, so this is kind of fun. So thanks, Webb, for having me. This will be a bit more of an applied side talk, trying to talk a little bit about the types of imaging that we're working with the data from in the lab and collaborating to advance these types of imaging. And so I work primarily in data science and computational neuroscience. And so the things that interest me are how do brains perform computation? And to do that, I think about the statistical structure of data. And my lab uses that both to try to design better data acquisition methods and to model that data to say, okay, we've recorded from 100,000 neurons in the brain. Can we actually say something about the computation that the brain is doing to make a decision or navigate the world? Or what is the difference between a healthy and disease state brain? And so a lot of this really depends on the data acquisition. And so that's what I'll talk about today, because that fits with the theme. And so one of the big things is we can see a lot more of the brain these days. We used to be able to just look at one neuron at a time. Of course, there's all this electrophysiology that's been going on to try to catch more and more and more. But imaging, and this is why I think this is important to a crowd like this, where I'm hoping to get more imaging people kind of interested and excited about this type of domain. Imaging has really provided a new avenue to see so much more of the brain at once beyond traditional electrophysiology. And so we'll talk a little bit about this. Just a little bit of rationale, why is this so important? Historically, we think of the brain as being kind of divided up into little areas, there's the visual cortex, the auditory cortex, LIP, PPC, and every time you go in, you can only record from one little area at a time. And so all our models of brain computation are area specific. But all these large scale recordings, for example, on the right here, the left, is a light sheet image of a zebrafish larva. So you can see, each dot is a single neuron across the entirety of the brain. And the color represents how much of the location of the fish in the world is being encoded, right? So I won't worry about the details of this experiment. Needless to say, there is a quantity that you can measure that tells you how much is that particular cell responsible for keeping track of where the animal is in the world. And you can see that these dots are everywhere, right? The red is everywhere in the brain. It's concentrated in one location, but it's everywhere in the brain. And so the ability to see more is going to change how we think about neural computation and how the brain performs tasks. So I will start with, if we're gonna go through all of this and it's still not gonna work. All right, we're gonna just move on then. So I will start with just kind of a brief overview of calcium imaging, right? So what I want to focus on is a particular type of imaging called calcium imaging. It is a raster scanning method. So that means that because you are trying to image deeper in the tissue, you are focusing light through tissue, right? So this is a non-linear optics framework, and you are essentially illuminating one point at a time and raster scanning back and forth throughout an entire plane underneath the tissue surface. And you're illuminating anything in that plane, right? So you illuminate, you're changing, you're stimulating with light, usually red light, and then you receive backlight. And so anything that intersects with that plane of imaging, you get the shape of, right? So in particular, right, what people do, and this is where this is 2-photon imaging. What people call calcium imaging is when you do this in animals where you've introduced a protein into the brain, either transgenic or virally, where you now have proteins expressed in every cell. When the cell fires, calcium floods into the cell, interacts, conforms with the protein, and that protein becomes fluorescent. And so what you end up seeing, and this is, I'm sorry, the video didn't quite work out, but you see these little flashes of light, little circles light up, and every time a circle lights up, that's a cell firing. So this has been pretty transformative in systems neuroscience. And there's a lot of areas in which we're still trying to take this imaging modality and make it better. And this is, where I want to convince you all that algorithms can really have a big impact here, and thinking about the type of stuff that's been presented so far, and other work that this community has been doing. Right, so there's a lot of questions about improving the quantity of the neurons that you're recording. In particular, computational imaging, I think, is going to be critical, and this is what I'm going to focus on today. There's a lot with improving the quality of data processing, right? So all of the types of denoising methods, using everything from simple estimation procedures to deep learning, is critical, because these images are taken in vivo while the animal's moving. There's a lot of artifacts, there's a lot of noise. There's a lot of challenges in just getting a robust signal out of them. And there's the question of imaging across scales, right? So one thing I'll mention is that the macro scale of single cells is very interesting, but the way that cells talk to each other, right, are even smaller, right? They're these spines, they're these little pieces of the cell that release neurotransmitters from one cell to another. And that's incredibly interesting, because that's how the neurons actually, that's the connections, right? That's the W matrix of the brain. And that's even harder to see. It's at a much smaller scale. And there's a lot of questions of doing things faster in real time, so you can make online changes to experiments and really probe the brain in more creative ways. So I want to start with computational imaging, and really thinking about making imaging either faster or larger, right? So there's this fundamental trade-off between that a lot of you deal with, which is there's the resolution of your imaging, there's the scale of your imaging, and there's the speed of your imaging. And those three trade off with each other in a fundamental way. So you want to image larger, you might not image at the same resolution if you want to stay at the same speed, and so forth. What's one way to kind of combat that is to design your optics in such a way that there's algorithms that can compensate for the deficiencies that you build into your optics, right? So you might image blurrier by imaging faster, and that would create a worse resolution, but maybe you can sharpen the image later in post-processing. More generally, if anyone has dealt with, I started my career in compressed sensing. If that's the kind, right? That's kind of like the extreme version of it, where you really are creating very encoded optics to compress data so you can take it faster or over larger areas, and then the algorithms compensate for that. I got into this a while back in this project that I won't talk about right now, but is a fun project called V-Twins, where we actually did stereoscopic encoding of volumes into single planes. But this was kind of my introduction in that you could do funny things with optics, in this case illuminate with a V instead of a point, and you end up with data that's basically a stereoscopic projection of a volume, and you can recover the individual objects that were fluorescing. So this particular project I'm going to talk about today is how to go faster, and this is in collaboration with GE, being led by Esther Wang, who has joined us. So Esther, please do correct me if I say things wrong. And we want to understand effectively how to increase the speed of this technology without sacrificing any spatial resolution. And there's two reasons why or two, I guess, use cases why this would benefit the field. One is, if we image faster, we can look at more types of fine scale dynamics of the brain. So we can go from imaging at 30 hertz to 600 hertz. Now we can go from seeing kind of general changes in the neural state to seeing like close to single action potentials in the brain. So single firing events. The other is that, right, we can actually have large optical windows into the brain. And so if you can go 20 times faster, you can use random access elements to essentially image 20 times more of the brain at the same speed, right? So this is, again, getting back to that trade-off. So what's our approach? Rather than doing this whole raster scanning business point by point by point across the brain, we're going to use an approach that we call neuroimaging by oblong random line acquisition, or NORA. And the first thing that we do is we modify the point spread function a little bit into this like little stretchy oval, right? So this is done very easily. This is a trivial change to any two-photon microscopy. You put a cylindrical lens in the path of the light. Like, this has been done. This is a very simple thing to do that you can just implement immediately, right? No, you don't need fancy optics. The other change is that rather than trying to force ourselves to take light from every single part of the image, we're just going to give up. And we're going to say, there's going to be areas that we just don't image on a particular frame, right? So keep in mind, this is a movie. We're taking frame by frame by frame by frame. If we don't take half of a frame, it only takes us half the time to acquire that frame. And so we're just not going to take the whole frame. So using the fact that the hardware element of this fast raster scanning in one dimension, right? So this is raster scanning. There's a fast dimension. There's a slow dimension, right? You go up and down really fast. We have resonant galvos that can do that super fast. Really what takes time is rastering in the second dimension, the slow scan dimension. And so we're going to keep this fast scan going. And we're just going to subsample the lines. So we're going to take less lines at every frame. And to compensate for the fact that we're losing so much information by not taking the full frame, we're going to do different lines at every frame. And so the optical change, right, the hardware part of this computational imaging approach is to blur and subsample. And what happens then, what we claim is that you can reconstruct your video from this data. And the way that what we use, right, that, remember, I like to think about statistical structure. We use the spatiotemporal statistics through a low rank prior, right? So this is going to be very direct. This is just using the fact that the pixels are correlated in space and time. So we're going to claim that this low rank prior enables the recovery of this highly undersampled area. And so just to kind of recap, right, we subsample and blur and reconstruct, and we want to recover using this low rank. And I should mention that this low rank prior is not something that we pulled out of a hat. This is a very classic way of denoising this type of data. So it is already a prior that's used in some form of inverse problem. And we're just saying that this is a very powerful prior. We know it's a very powerful prior because there was a whole field of matrix completion using this to fill in incredibly undersampled matrices, right? If anyone remembers the Netflix prize from a couple years back now, or maybe I'm just old now. And so what is low rank? In case this is probably something that most of you have already seen, but just in case, right, here's the French flag. It is low rank. That means that every row is essentially the same as every other row. This is a rank 1 flag. The Maryland flag, not low rank, right? You cannot just replicate the same row over and over and over again and recover the flag. And practically what this means is if you go ahead, and I do not recommend doing this, and you cut pieces out of the French flag, you could easily fill it back in. If you cut pieces out of the Maryland flag, also probably don't do it. It is much harder to fill it back in, right? This is the whole principle. So practically for us, we're using the fact that this is not recovering image by image the video. People try to do this, right? And people can do this, and you can throw all kinds of image recovery stuff frame by frame at this problem. You are doing yourself a disservice by not leveraging the temporal correlations that are incredibly strong in this data. And why is it incredibly strong? It's because you're imaging the same tissue frame after frame. And so as you go from frame to frame, the same pixels are the same object through time, causing these bars that kind of look like the French flag. And those bars don't change location. And so you can cut pieces out and you can fill them back in, right? Much like what we did with the French flag. This is it, right? It's a little bit more complicated because the intensity of these bars is changing slowly over time, but the rank, the effective rank, right? It just means it's not rank 1, it's rank 2, 3, 5, but it's still much lower rank than the number of pixels or the number of time points. So how do we recover, right? The forward model here is super easy. It is both both blurring and subsampling are linear operations that are incredibly cheap to perform. And we want to find the best video, right? We're performing video recovery, remember, that matches the observed measurements. It matches the blurred lines that we saw through this forward model of blur and subsample, while having as low a rank as possible. And we're going to start with the simplest possible thing ever. Well, okay, not the simplest thing ever. The simplest thing that has these properties that we know a lot about, which is nuclear norm minimization. To test this, we need to be able to compare to some sort of ground truth. And this is really hard to do in reality, right? We could build the system, we could test it, but we wanted to make sure that we could really recover data that had the right statistics before we went ahead and tried to like build multiple systems with interleaving optics to get the right ground truth. So what we leveraged is we leveraged a piece of work that I had done a little while back called the Naomi simulator. So the neural anatomy and optical microscopy simulator. It is a biophysical simulator, so it builds in silico tissue, vasculature, cells, dendrites, axons. It grows them all using essentially anatomical information that we've collected over the decades. It creates activity for these cells based on biophysical models of calcium interactions and action potentials. It simulates the light through scattering tissue, right? These are all biophysical models. We're not sampling from the forward model is what I'm trying to emphasize here. And that creates highly realistic images that have been used through, I've been happy to see that people are using this to test a number of different imaging methods by building in different optical paths and testing out different recovery algorithms all on the simulated data. So how well does this work? We simulated Nora in the Naomi simulation. This is playing, right? Oh, okay. And the idea, what should be showing here, it should be a full video. Oh, there, that shows a little bit better. So we took 1/10 of the number of lines, right? So we are imaging effectively the simulated imaging, but at 10x speed, right? So you can basically take 1/10 of the amount of information for every frame. And what we compared here is for individual cells, there's activity over time. And we compared in blue, which is the extracted activity from the recovered video using matrix completion from this blur and subsampled, to the ground truth that's provided by the simulator. So the simulator used activity to generate the data. And we're saying that we can recover it using 1/10 of the number of samples that you normally take per frame. We've pushed this to 1/15. Oh, this video plays a little bit better. And you can kind of see that there's both on the ground truth and the reconstruction, the same cells kind of light up at the same time, assuming that it keeps going. Well, hence we have the line plots too. The line plots also show the ground truth and the now imaging 1/15 of the number of lines can still recover. So we've taken this a little bit further. We just put a preprint online about this method literally last week, and we've taken it up to 1/20th already. So we're seeing just how far we can go with just a simple nuclear norm minimization on a blur and subsample scheme. And it seems like already, it's really panning out in terms of being able to leverage the full spatiotemporal statistics of the data. So why not deep image? Why do we do this low rank thing? Well, from the onset, we wanted to see just how far we can go with it, right? So low rank is itself a very powerful prior. Matrix completion has theory. It works really well. And in fact, we're adapting some of the theory from prior matrix completion to this particular imaging paradigm. We needed 0 training data. So we could point this at anything that we wanted as long as it had this low rank structure, and it would work. The other thing is that the systems neuroscience imaging is a bit of a wild west in terms of a lot of people build their own microscopes, they trade out components, everyone's imaging different animals under different conditions with different stuff, and that creates a really difficult out of distribution problem. So I've tried this myself. I've taken really well-established papers published in PNAS and Nature Methods, and I download their pre-trained networks and I try to adapt it to data, and it does not work. And then I do low rank and it works. So for all of those of you studying out of distribution data, here's an extreme case where almost every lab is out of distribution from any other lab to the point where it really damages this generalizability. So that is, I'm gonna not say that, but because we're at 10, but I will say that this is one imaging method that my lab works on. I'm kind of a data junkie, so we do a bunch with optical imaging. We've started doing functional ultrasound of the brain. So I was happy to see a lot of ultrasound stuff. There's a lot of open problems in neuroimaging at the systems level. And it would be fantastic to get more people coming in and bringing kind of advanced techniques. And I think that there's a lot of benefit that could be had here. So with that, I would like to thank my lab and thank my funding sources. We're thinking of you. And then NIH, NSF Career, the Chan Zuckerberg Initiative, and the Rayner Sarabella Project, which kind of got squished over here. So I'm happy to take any questions. The preprint is online right now. And this is my website, and my office is down the road if anyone gets bored. So thank you.
Perhaps I'll start off. So this data acquisition you're doing, where you're kind of just taking this banded data, but my understanding is you've intentionally added this blur kind of across bands, right?
Yes.
And I'm guessing the motivation is that gets you some information about the stuff that you're not collecting, but it's kind of blurred in. So how do you decide how much blur you're gonna put there?
Yeah, that's a great question. So far we've just tested that the blur actually helps by testing with and without blur. And it does, but it's true that optimizing that blur is going to be in a useful place. And so that's one of the things we're hoping the theory actually points us to is, right, there's going to be, right, so the way that these theories, these kind of sampling theories work is that there's going to be a coherence term that tells you how similar is a single measurement, which in this case is a single point spread function, with the objects that you're imaging. And so at a certain length, it's going to start, that coherence term is going to start having a negative detriment. And so we're hoping that exact term is going to tell us like what is the range of values. Right now we've mostly been picking like 3-ish microns, right? So it's a 3 by 1 oval if looking at it from on top. But that, again, right, we just picked a number.
Talked about low rank, I thought in time it might be changing a lot, but in space it's the same cells, so there might be the spatial component might be changing a lot less, so much stronger correlation. So if the rank is different in space and time, and this leads me to the blur, right?
Okay, So I think the way to think of it is that the matrix that is low rank is pixels by time, right? So it's really that the same pixels are in the same location, and sure, their activity is changing over time, but their activity is, so you think of the area around one cell. You have lots of pixels, but all the pixels inside the cell have the same trace, right? So they are effectively rank 1. And so there's a relationship between the number of independent fluorescing objects and the rank. And so if the number of fluorescing objects is much lower than the number of pixels, that's immediately low rank, not even accounting for the fact that the cells themselves are correlated with each other in terms of their activity, which lowers the rank even more.
Yeah, thank you. I think the connection that I was thinking was in terms of making like if you could make the blur function pretty big
Yeah.
and allow you to go faster in time.
Yeah, so this is the other challenging part of multiphoton microscopy is that the dispersion of light through tissue means that if you try to spread your point spread function out too much, you basically get no signal. And that's so I would have loved to be able to do like Fourier measurements. That would have been great. That would have fit perfectly with my background. We can't do Fourier measurements. Like those kinds of things. Yeah, but it's a great point. It's a great question.
Maybe one more quick question. So you're very focused on getting this really quick data acquisition method. How important is the other side of that? That once you have all that data, is it okay to process all day long, or does it need to be processed quickly and available immediately?
Yeah, so typically it's okay to have it run overnight or something if you needed to. It would be great to do it faster because it lets you online get a better sense of what you're looking at, right? The downside is that, right, you don't get a traditional image, so you can't like look at it and say, okay, where am I in the brain or something like that. So usually like you would have to take something like traditional images just to like localize yourself in the brain. And then, but yeah, in terms of processing speed, like there could be a time lag. It's not like there's a patient waiting for something today, right? So time could be. Now we're still interested in making it run faster, and I think that's where kind of these implicit priors like could be a useful area. We haven't touched that yet. We just wanted to make sure it was possible first. That's a great point.
Well, thank you so much, Adam. Let's give Adam one more hand.